APJ Abdul Kalam. Let's begin our session. I am Vidya Vijayan of 2nd PG Biotechnology. It's my privilege and entitlement to welcome you all to this international webinar on the topic Advanced Genomic Technologies in Healthcare by research person Sir Sanal Kumar Subhadra, who is working as a research scientist in the Lausanne University Hospital, Switzerland. I would like to welcome our Reverend Principal, Dr. Sister Asha Therese, our respected HOD of the Biotechnology Department, Dr. Vijay Mary Varghese, Director of Research, Sister Floret, students, and all the teaching faculties of the Biotechnology Department and other departments of St. Joseph's College in Yalakuda, and also participants from other institutions. Once again, I welcome each and everyone to this international webinar. Now, I request Dr. Vijay Mary Varghese ma'am to deliver the welcome address. Please, ma'am.
Good afternoon, everyone. It's with immense pleasure that I would like to introduce and welcome our today's resource person, Dr. Sanal Kumar Rajendran, who is the resource person for the international webinar jointly organized by Research Center and Biotechnology Department. He has completed his PhD from Rajiv Gandhi Center for Biotechnology, Trivandrum, and did his postdoctoral fellowship in University of Wisconsin-Madison. And currently, he is working as research scientist at CHERV, Lausanne University Hospital, Switzerland. He has got more than 60 international publications to his credit, and his expertise lies in the fields of stem cell culture, stem cell biology, stem cell differentiation, molecular biology, epigenetics, and developmental biology. I'm happy that we have got such an eminent and expert resource person for today's session. A very hearty welcome, Dr. Sanal Kumar, to this session. I also welcome all our dear participants and my dear colleagues to the session. Thank you.
Thank you very much, ma'am. Now, I would like to welcome research scientist Sanal Kumar sir to take over the session. Sir, please. Sir, I believe there is some technical difficulty. Sir, are you there? Sir,
There are some
am I audible?
technical issues. Sanal sir will be joining soon. There are some technical issues. Please wait. Sanal, can you hear me? Okay. Okay. Hello. Actually, we cannot hear you. Maybe you are muted. Can you please unmute?
Actually, I cannot connect through my institute ID. There is some compatibility issue. I don't
Okay.
know really what's happening. So what should we do? Because I don't have the
If
slides.
you just email me your slides, I can present from here, and you can see the presentation there also.
Ah, that's cool. Okay, just a moment. Give me a minute. It's perfect. Okay. I'm extremely sorry for this.
Okay, that's okay. That's okay. No problem.
Um, Actually, we got a very complicated security system because it's a hospital and it's have a lot of patient information. So
Yeah.
they have set it like, it cannot download any software or anything,
Yeah.
only through the lab system.
Mm-hmm.
Okay, forward. Nigel. Okay, you can go ahead and introduce if you want, then so that
Yeah.
we will be. I'll just forward the. I'll keep my mobile here.
Okay. Uh,
Yeah, something like
Genius,
this.
can you introduce him once again? Actually, we thought you are in, so we.
No, it's okay. If you're done, it's fine. No
No,
problem.
no.
It's okay. You can just go ahead. Okay.
He didn't see it, maybe.
Yeah, the presentation
Yeah.
will be. I will be showing the presentation. Once I get the presentation, I will be presenting.
Okay.
Yeah, meanwhile, I can introduce you once more. I can welcome you once more at least.
Yeah.
Really sorry. Okay, please go on.
Okay. Okay, good afternoon, everyone. It's with immense pleasure that I would like to introduce and welcome Dr. Sanal Kumar Rajendran for today's international webinar jointly organized by Research Center and Biotechnology Department. He has done his PhD in Rajiv Gandhi Center for Biotechnology, Trivandrum, and did his postdoctoral fellowship in University of Wisconsin, Madison. And now currently he's working as a research scientist at Lausanne University Hospital in Switzerland. He has got more than 60 international publications to his credit. And his expertise lies in the fields of stem cell culture, epigenetics, molecular cloning, cell signaling, molecular biology, and developmental biology. I'm happy that we have got such an expert and eminent resource person for today's session. A very hearty welcome, Dr. Sanal Kumar, for today's webinar.
Thank you. Thank you very much.
Okay. I also welcome all the participants and my dear colleagues for today's session. Thank you.
Okay, Nigel, did you receive the
Yeah.
presentation?
I just got it. I will present it now. Okay. Yeah, got it. Let me download.
Yeah, it's like only a few slides, so won't be complicated. For my sake, I'll have the slides here, so it's not a problem.
Mm-hmm.
Maybe I'll just start, and then you can, once your slides are in,
Yeah,
you can just.
you can start. Now.
So thank you for the invitation and for the nice introduction. That was great. So I'm extremely sorry for this delay and technical issues because we have a highly secure system, and this is not. We are not used to Google video chatting. We usually use Zoom and other softwares. So I'm extremely sorry for the delay. So let's start with a little bit of introduction. Most of the things that I'm going to talk today is related to genetics and genes and things like that. And most of you have learned the basics in school, and probably now you're learning. I guess most of you are like the bachelor's and masters, so it's not complicated. It's a very simple, very fundamental questions that we are trying to address in our lab. I'm not going to present you a lot of data, so don't worry about that. I'm not going to bore you. Just some of the basic concepts that we are trying to study in the lab, and to give kind of an introductory to how the research is being done in such fields, especially with the unfavorable situation right now we are facing with the coronavirus. And most of you know the efforts that a lot of scientists and labs and clinicians and physicians are putting together to have a effective treatment strategy or vaccine. So something like that, we are trying to understand the biology of the disease. Okay, there you go. I have these slides.
One second.
again issue?
Yeah, it's opening. Say it's opening, but it is not opening. Really
Oh,
open.
no, it's not. Actually, it's a very simple slides. It doesn't have a lot of slides. Should be fine.
Okay, got it.
Got it?
Yeah. Yeah. I hope the participants can see the slides now, right?
I cannot. I can see your picture.
It's presenting. It says it's presenting. Anyone,
I don't know.
can you see the presentations now?
No, sir, it's not visible.
I think it will be visible soon. It's presenting. I'm presenting now.
No, it's it's just your picture. Mm-hmm. Or should I do my front back camera or?
No. I will presenting once again.
Yeah, there you go.
Okay.
Okay. Okay. So maybe I'll just say that the next slide, then you will move, right?
Yeah, that's perfect.
So this is this title. Okay. The next one, we'll just move forward. Let's see. Okay. Just before going to the details, I just wanna say who we are. Actually, this is the place I work. It's called SHUV. It's actually University Hospital of Lausanne, and it's a French. Actually, it's a French. That's why you see the French. It literally says that's the University Hospital of Vaud, which is the Lausanne region, and it's a medical school, and we have the research units, and I am part of the Pathology Institute, and most of the labs here work on some more or less something related to clinical, work, and we are like my immediate PA, and our lab is core is working on cancer subtypes in children, and we're trying to understand the biology of tumor, mostly basic biology, and we have some clinical studies as well. So I'll give you a description later, but this is a place we work on. And the next slide, please. Okay, this is just an outline of what exactly we're trying to do, but I will come this later so that it's easy for me to explain. Next one. Okay, here actually, as I was mentioning that I will be mostly talking about genes and a little bit of genetics and genome. I hope everyone is familiar with these words like genome. At least from your school and +2 and the bachelor's degree, you probably would have studied a little bit of molecular biology, fundamentals of molecular biology. Uh, just try to refresh your memory. Like from the picture, you can see that the structure of DNA, the double helix, and then you can see something called gene. And this, what is a gene? It's basically the transcriptional unit. It doesn't have any borders as we see here. It's a DNA sequence, but we know that the gene has specific structures like introns, exons, the promoters, and UTRs region. All these are like very fundamental things. I'm not going to explain any of these, but you know that we have this basic composition of the DNA structure, the genes, and then we have the histones and the nucleosomes, and then it comprises the chromosome. And depending upon the organism, we have different number of chromosomes. As we humans, we have 46 chromosomes, like 23 pair. So why I'm saying all these is like that the term genome referred to the entire genetic information of an organism. So for example, in human case, in our case, we got these 46 chromosomes. And if you take all the DNA, everything together, complete information, even including the mitochondrial DNA, that's called the genome, everything together. Okay. And genomics is something like, it's studying the genome. Simple. It's not complicated. It's very simple. If you study something related to gene or genetics or the entire genetic information, it's called genomics. And as any other branch of study, we need tools and techniques to study the genetics, because genetics is one of the oldest fields of study in biology, starting from the Mendelian era. But with the right tools, because it's very difficult, it's like a very minute structure and it's very complicated. We need the right technology to understand the whole thing. It's very, very complicated. As you imagine, we have 23 chromosomes, and that 23 chromosomes comprises almost 3 billion sequence reads, or 3 billion sequence base pairs of information. 3 billion, which is like a lot of information that we cannot process, like a human brain cannot process everything together. So we need additional help from the computational field, or programs, or kind of techniques to analyze the whole information in a large scale. So we have in the previous days, we have studied the chromosomes, its abnormalities, the structure of the chromosome, then down to the genes, and we later we have the sequencing. We acquired the sequencing technology to sequence the bits and pieces of the genome and understand the genetic information. Even though not completely, but later the sequencing technology became more advanced and used the computer or computational biology incorporated for the data analysis, and then we have the complete genome project or human genome project, which means that we sequence the entire genome of an organism. Initially, we sequenced the small organisms like bacteria and viruses, like which has a very small genome compared to human being, so it was easy to study. Then we've been sequencing the human genome, and then we have the whole information of the genome, the entire genome. Now we know how many genes we have, and what is the genetic complete makeup of the genetic makeup of a human being. Also, we have sequence from different races, like we have white. We have Asians, we have African Americans, Indian, or any population you take, we have different variations in our genome composition. So all these things actually comes under genome sciences or genomics, studying the genetic information of an organism. And I will be mostly talking about human. I know that you probably would have botany students here, but most of my expertise, and I'm trained in human biology. But down to the chromosome and genetics level, there is no difference between human and in plants or any other organism. The basic composition, the mechanisms, and the organization of the chromosomes and genome is exactly the same. Okay, next slide. So, important question, why do you want to study the genome? Of course, we want to know what exactly, what is the genetic composition of an organism. But other than that, in terms of health sciences, we always say bench to bedside, which means that scientists or the clinicians or physicians do research to identify any drugs or any treatment strategy for a given disease, you know. So just talk about this aspect right now, the healthcare issues, why do you want to study genome? Other than, people study genome to know to about the genetic, the basic biology, to study evolution, and other aspects of biology. We need to study genome, but just to imagine the reason, the healthcare, the benefits of genome studies or genomics in healthcare is tremendous. It has given opportunities, like for various disease treatment strategies that we have developed and being developed. So it's very important, and it has helped us to understand the biology of the disease, either the pathogen or the disease itself. And then we need that information. We need the information to for the better treatment strategies or drug development. So I'll give you a couple of examples, and can you just click? There should be another. Yes. Okay, so I'm going to give you the very suitable example, like the COVID situation. We have this SARS-CoV-2 virus structure here. Everyone is familiar this one or other versions of the coronavirus almost every day in our lives right now. So as you know, that's a single-stranded RNA virus. So as soon as it was identified as a virus, as a coronavirus, what people have done, people means the people in the scientific field or the drug companies have done, is try to sequence the genome of the virus. They just took the entire genetic material, and because it's RNA, it has to be reverse translated, transcribed into DNA, and then you can easily do that and then sequence the entire genome. So once you have the genome, if you see the picture on the downside, you can see something labeled as RF1, and different weird names, which are the genes, the corresponding genes of the coronavirus. So I'm not talking about anything about coronaviruses, because the reason we want to study, one of the first thing that the scientists did is to sequence the genome of the virus, because we want to know what exactly the virus is. We want to know the biology of the virus. We want to know which class it is, and in evolutionary history, which is the closest organism it is. And then we want to identify a unique region. Unique region means a specific region to develop vaccine, because we know the virus. How does most of these viruses behave? But we want to know, if you want to develop an effective vaccine or a treatment strategy, we want to know the exact precise genetic information, or the structure of the proteins. The spike proteins are most of these days people are talking about spike protein, because most of the vaccines are being developed to targeting this region, is mostly unique to the virus. Anyway, so this is one of the reasons. If you don't have the capacity to understand the sequence or the genome of the organism, of this organism, it would have been taken ages to do something. Now the vaccines are under trial, and everything, and it's there on everyday news. So you have more information than me probably. So this is one of the applications of the genome studies. The technology has improved so much that we can sequence the entire genome like in hours, like, or minutes these days, like especially genome like this very small genome, we can sequence it very, very easily. Then we have the computational power. We have the bioinformaticians, or it's called computational biologists, who analyze the data and tell, like, okay, this is a region which is unique to this virus, and this is a region which is optimal for the vaccine development, a targeted therapy, or any potential drugs. We have different types of research going on based on the genome information. That is one application of studying genome, okay, or genomics. Next slide, please. And this one is another example of a cancer that mostly we are studying in our lab. That most of you know that normal cells give rise to cancer cell. I know it become a cancerous cell because some of the signaling pathways or some genetic alterations that happens in our DNA. Most of the cancer is triggered by a genetic alteration. You have mutations, you have chromosomal abnormalities like translocation or duplication, deletion. You could have any of these chromosomal abnormalities or mutations of a given cell, which can give rise to cancerous cell. And there are more than hundreds of or different, I don't know, hundreds of different types of cancers which has different genetic insult, which means genetic mutations or triggering mutations, whatever you call. So if you want to study why why the cancer is happening, or what is the current situation of the cancer cell, because you cannot treat cancer with a single magical drug, even though some people claim that in our place. We cannot treat any disease with a magical drug. Especially cancer is not a one disease. It's like a collection of several diseases. So for each and every cancer type, you need to understand the biology of the cancer. You should know what exact type of mutations the cancer has, or what type of genes are not functional, or what types of genes are like over acting. This kind of a lot of information you need to know about before making a treatment strategy. Otherwise, we can just do a classical treatment like radiotherapy or chemotherapy, which are not specific targeted therapies. It can be done for any cancer, but for any specific cancer treatment, you need to know the biology of the cancer. So here is another application of the genomics comes in, because we want to study the cancer subtype. So we take the cancer, we take the tumor cells, then we study them, we study its genomics, and then we know how different is the cancer cell is from its counterparts of the normal cell. And then we know what is happening in that cancer cell in terms of signaling, in terms of mutated genes, in terms of overexpressed genes. All these information you get only from the studying the complete or entire genome of the cancer cell. So even though the cancer cell is originated from a normal cell, which has a normal genome, like in the picture you can see that it has a controlled growth, it has a precise function, and everything is controlled in a cell. But when you lose the control due to some of these insult, then it become uncontrolled. So we need this information, and the only way to get information is to study the genomics. You can study the biochemistry, of course, the physiology, the pathogenicity. You can study many things, but to get the complete picture, in terms of the genetic information, or in terms of the expression of genes, down regulation of genes or up regulation of genes, and or any protein function, anything. And if you take everything in the entire genome wide, that would give you a complete picture. Then previously we used to study like one gene or a few genes, things like that, but that's not complete picture. So genomics comes in play here to get this information as well. So these are the two major types things that we can we utilize. I mean, we have several other applications of genomics in healthcare, but just to give an introduction, I mean, this would be a couple of examples that the useful of genomics in healthcare. Next slide. Okay, so in talking about genomics, you know, I don't know whether you have studied, you have this in your syllabus. I have no idea about the current syllabus. So when we were studying, we didn't study any of these things, but maybe it has changed. So when you study about genomics, you know, you can put them in two categories, like the structural and functional. As you study in most of the things, we have a structural component and then the functional component. Actually, I talked about both these already. So what is a structural genomics? It's just basically a structure to study the structure of the genome. Structure means just the composition of the genome, which is trying to understand the sequence. So, for example, in the coronavirus example, you take the coronavirus and sequence the entire genome, and you have the physical sequence of the genome, which is the structural genomics. You are not studying what the genes are doing. You are not studying, you know, any of his proteins function or anything like that. It's just the information of the structure of the genome. That is structural genomics, just sequencing. Then try to understand the mutations. You know, you keep saying that the coronavirus is getting mutated, you know, or the virus that we have in India is different from the one we have here in Europe or in US. The different parts of the world have different types of, you know, they classified like virus A, subtype A1S, or I don't know. I'm not an expert in virology, but still. So how do they do this kind of subtyping? How do they classify different types of viruses? It's based on structural genomics. You take the virus from different regions or different patients, and then you sequence and then you identify mutations or variations. You can call it either mutation or variations. Like, you know, only some parts of the genome will be like similar in India, but they are not same in the European strain. So we call it different strains of the virus. So to call these different strains of virus, you should know how you should know the sequence of the organism. Similarly, when you take the example of cancer, you take a normal cell, you sequence it, and we have the complete genetic information of a normal human cell. But you take a cancer cell and then you sequence it. Then you identify all the mutations that cancer cell acquired during the process of cancer. It's not one or two. Depending about the cancer, we have hundreds of mutations we acquire in our genome actually. So you will get the entire information. You'll get all the information of all the genes or non-transcribed regions, any type of mutations like single nucleotide polymorphisms, SNPs, or deletions, you know, duplications, translocations, inversion, any type of genetic alterations that has happened to the cell, we will get that information. So this is actually structural genomics, you know, to take the genetic material of an organism, to sequence it, identify its makeup, and compare it with a normal counterpart. For example, in the disease cell, we compare it with a normal cell, or for the organism, we compare with the, you know, similar organisms, and say we can easily classify which evolutionary group a tree it belongs to. So these type of studies we collectively call structural genomics. So next slide. Okay, so this is the one I actually explained. Actually, you know, you do exome sequencing is the oldest technology, just sequence the exomes. Then you can also do an SNP array, which is like just to identify the SNPs, like the small single nucleotide polymorphisms. Then you can do the whole genome sequencing. And nowadays we are doing whole genome sequencing because the whole genome sequencing is easy. And now it's not super expensive. So, you know, it's anybody can do it. It's not a problem. Then you have the personalized medicine and gene therapy. These things are like sophisticated genome applications, like, you know, because each person can be treated with their own genetic information, because each one is different. Even though we say that the human genome, and we know the normal human genome information, each and everyone has their own sequence makeup, you know. somewhere or the other places we have differences in our genome. So to identify that information, you need to sequence each and every one of us. So for example, if we have got any disease, then they take our DNA, they sequence it, then they identify any specific mutations or sequence variants associated with us. Then based on that, we can predict whether the existing treatment is optimal for us or not. That can be done, and this can also be used to identify the person, like in criminal cases, like DNA fingerprinting or DNA match. You probably would have seen this in the forensic movie, forensics, right? The Plavino movie. So something like that. It's super fancy movie, but still, it's possible that we can easily identify the person, because each and everyone is different in its genetic composition. And gene therapy is another application where we I don't know, the latest Nobel Prize is announced for a technique called CRISPR-Cas9. Probably you heard about, and someone would have talked to you about this one. So it's one of the gene therapy type of applications. I'm not going to the details, but any type of this kind of gene therapy applications, the basic understanding is that you should know the genome. You should know the genetic information of the organism or the disease. Then only we can go and correct it. If you find a genetic mutation of a person, if you want to correct it, you want to know the information, right? So these things comes under the applications of the genomics, and we can call it the structural genomics or even the functional genomics. It's not like super independent. It's completely dependent one and other. So next slide, please. Yes, functional genomics. It's nothing different actually. So you take the organism, you sequence it, you have the information of the information of the genome, and then the sequence and any mutations or anything. That's good. That is structural genomics. Then based on that, we can predict. We can see what could have gone wrong. Okay, this gene is mutated, so which means that gene is not functioning, or that gene is mutated in such a way that it's overacting. All this kind of thing we can predict from structural genomics. But to really see what is happening, we want to know what is happening. Like so genes is just like the DNA. It's the piece of DNA in our nucleus or in our chromosomes. What it actually does, it has to translate its message to one form or the other form. We call it as basically gene and gene and gene, but gene is just a physical thing. It's there, but it cannot go and act. It has to translate. It has to send its message in one way or the other way. To study this one is we call functional genomics. The function of the gene, of the genome. How do we study the function of the genome? Okay, we know the sequence, we know the mutations, and we can predict and we can say, okay, this could be happening, this could be happening, blah blah blah. But how do we study the actual function of the genome? That comes under the genomics. Next one, please. So as an example, to put everything together in a very simple question, I've been talking about genome and genome and genome. So we know that almost everyone has developed from a single cell, the zygote, right? 50% of the DNA from father, 50% from mother, and then we have the zygote. And from a single cell, we develop the whole organs. We develop the whole organism. And if you talk about the genetic composition of an organism, if you take a cell, like in the picture, you can see that we have got different types of cells. If you take a neuron, muscle cell, cardiac, or any other type of cell, the genetic composition or the genome is entirely exactly the same, photocopy, no difference. There are exceptions that you study, I know, but most cases, the genetic composition or the genome is like exactly same. But these cells are doing different jobs. How? This is one of the fundamental questions people had in the early days. Like, you know, we have exact same copy of the genes. It's not something like that, okay, you have only neuron specific genes in the neurons and muscle specific genes in the muscles. No, we have everything in exact same order, exact same sequence. Everything is like photocopy. But how does each cell is looking different, they're functioning different, their smell, their, you know, their taste, everything is different. So how does it happening? So there is the magic happens. Actually, the tissue specific expression or the cell type specific expression of the genes. So the reason I was trying to put this together is like to introduce some terms. I don't know, maybe you have already learned these things, the basics at least. But to put the idea of functional genomics, I mean, this is one of the best examples that I can put together. So if you take a neuron and if you take a cardiac cell, then you compare the genes which are expressed in these two types of cells, you know, you will see that majority of the genes are different. Though we have similar, you know, same type of genes are expressed in both cells for example, genes which are important for the basic metabolism, you know, energy metabolism, structural components, cell makeup, you know, those things are probably the same genes, but it has neuron specific genes are only activated in neurons and muscle specific, cardiac specific genes activated in the heart. So how does it happen, and how do you study this? We know that the genes we don't we if we sequence, we don't know whether the gene is active or not. We will only know whether the gene has any mutation or not. We don't know whether it is active or not. There is no, if you look at the microscope, if you look at the sequence in the sequencing machine, you don't see, you cannot tell whether the gene is active or not. You have no idea, absolutely no idea. For that, we need to quantify. The next slide, please. It's messenger. It's messenger. And you know this central dogma in the molecular biology, like we have got the DNA, that's the information. Then it's translated into mRNA, or different types of RNAs we do have, but basically mRNA. That is a messenger. Then that mRNA is again translated into protein. So DNA transcribes to mRNA, mRNA translated into proteins. This everybody knows it. Then how do we study the functional genomics? Is that if we want to know a particular gene is active or not, we want to quantify its RNA or the protein. So the easiest method is quantifying RNA. So to measure the transcript, to quantify the transcript, and it's called a transcriptomics. You know, or you can simply say that RNA sequencing, like our DNA sequencing, you can sequence the RNA. I'm not talking about the technique. You probably know it, but you take a cell, like the example I was mentioning. You take a cardiac cell and a brain cell, a neuron, and then you isolate RNA. Excuse me. And then you take the whole RNA and you put it in the sequencing machine, and you can sequence it. Then that will tell you all the mRNA molecules which are expressed and with quantity. Like it's a quantitative method, like, you know, you can quantify how many of this particular gene is activated compared to the other one. So I mean, it's completely quantitative analysis. You know the exact information, how many molecules of each RNA is expressed in each cell type. So you can also do the proteins. It's like protein sequencing is much more complicated, but you know, there are different techniques you can do. I mean, you have probably studied all this protein quantification, protein sequencing techniques like MALDI and blah, 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 all these things. But I'm talking about mostly the genomics. So this focus on the mRNA side. So this is one aspect of functional genomics. You know, we quantify the output of the DNA transcription, the mRNAs. Then that will tell you the exact function of how does the function of the particular genome is. You know. Then there are two more things that I want to talk about based on the previous slide. The reason why only the cardiac genes are active in cardiac tissue, and not blood genes are active. So which is called the transcriptional regulation. The process of transcription is regulated, and that process is called transcriptional regulation. There are different mechanisms, and this one of the mechanisms is epigenetics. I hope you heard and studied about epigenetics. Next slide, please. Okay, so this is something like, an example of how can put the epigenetics together. So you have the chromosome, you have the nucleosomes, and you know that our DNA is not just the DNA. If you talk about the chromatin, it's not just the DNA. It has it has the histones wrapped in it. As you can see the pictures, it's wrapped around the histones. And the histones are proteins, basically. And it has, these proteins can be modified, like a post-translational modifications, we would say. That's the reason I want to say this is like that, we don't have naked DNA. Even though you say that the DNA is like the double helix structure, it's not naked. It's like we have got the histones, and these histones can be modified. And the DNA can also be modified with methylation. So I'm not like talking too much about the epigenetics, but the reason is like that. This is a key for the transcriptional regulation. Okay, the epigenetics is the key for the transcriptional regulation. For example, if you want to know whether a particular gene is active or not, this process can tell you, whether the gene has to be on or off, like a switch. So depending upon what type of switch you have, you can have different modifications, and that tells you that actually, so for example, and I'm trying to put this in a simple way to explain. So for example, you need cardiac specific genes only in heart cells. So those genes has to be activated. So there are mechanisms to keep these genes on with the help of epigenetics. So we have these chromatin modifications so that only those genes which are required for the cardiac function will be turned on in that cell type, but not on any other cell types. So if you take the same genes, only those genes which are cardiac genes will be activated in some sort, activated with this epigenetic mechanisms. And with same epigenetic mechanisms, you can put the gene off, turn off the gene in any other cell types. So it's like a switch. It's just basically just imagine there's a switch that you can turn on or off depending upon where you are. Okay, something like you have lights in our house, or different light or fan or things like that. If you enter, you only turn on switches of things which you require. Fan or lights. You don't need lights in the morning. Like you need only lights in the evening or nights. So you can turn on or off, something like that. You can turn on or off the genes when it is required in different types of cells. So taking all these together, the mechanism is called epigenetics. Okay, maybe you have studied more than this one, I don't know, but just keep it simple. So this decides whether or not a gene has to be expressed or not. Okay, and this is how the specificity is acquired. This is how the same genetic information is regulated, like, so irrespective of the sequence, we have the same sequence, but the sequence doesn't matter. What matters is the epigenetic mechanisms to decide the deciding factor, whether the gene is activated or not. So that is important. So these mechanisms, the transcriptional mechanisms or the epigenetics, we can study using the genomics techniques. So this is one of the major applications of genomics right now, and we can test we can identify the epigenetic mechanisms, transcriptional regulatory landscape of the entire genome of an organism or of a cancer cell or any other disease cell, and we can easily say that okay, it's not just the genetic information. We got the genetic information from this structural genomics, but we now we have the epigenetics information or the functional genomics, and we can tell that okay, these are the switches are on or these are the switches are off, and based on that we can identify its activity, and then we can study further mechanisms as well. So this is one of the major fields that we study in our lab. To be precisely, this is the reason I want to talk about a little bit of epigenetics. So, and one of the major applications of functional genomics as well. Okay, and the next slide. I'm not talking about this one right now. Probably, I don't know how time is now. Yes, already we are 1 hour past. So, I'm not talking about this one. That's okay. So the idea is that just to give maybe a couple more slides, please. Yeah, this one. So just to give an example of what we are studying, in our lab, you have we have this normal cell. Then the cell will get some sort of mutation or oncogenic activity. Then it changes its fate. Then its epigenetic regulation will change, which means the types of genes which are expressed will change, and its morphology will change, its function will change. Then it will acquire additional mutations and oncogenic signaling pathways, and then finally it becomes the cancer. This is the process of tumor, the cancer generation and progression of the cancer in any cell type. So this is what we are studying actually in our lab. Okay, next slide. And oh, this is what exactly I told you. Maybe we can switch. We can skip this one. Yeah. So how do we study this? Actually, in terms of functional genomics or in terms of experiments, how do we study? So we take the patient sample, like the tumor or the cancer, whatever the type. Then we sequence them, the tissue, and we identify the any mutation. We first study how does the genome looks like, structural genomics. Then we know all the mutations, all the information on all the changes that happen to the cancer cell from the normal cell. Then we have that information, which is good. Then we also do the functional genomics, we study the histones. We study the other transcriptional regulatory and epigenetic modifications that could happen the cancer cell using specific techniques, genomics techniques. Then we have a complete picture of the cancer cell, like in terms of genetics, in terms of structural and functional genomics. Then based on that information, we either try to understand its basic biology, or we will also doing some sort of drug screening experiments based on the information we get from the studies. To put things very simple, this is how the lab functions. Not our lab, in most of the cancer biology labs in the world is working like this. So to get the patient sample, use genomics techniques to get the structural genomics, and then also most people work on structural genomics, but we also work on functional genomics. And then either try to understand the disease mechanisms, or develop treatment strategies, drug screening, or identify new drugs or things like that. Okay, next one, please. Okay, now we have the first slide. Actually, this is the workflow. So you can see that we have the hospital, we have the patient, then we isolate the tumor types, and we put them in culture in vitro. And then the red part is the most of the things I'm doing, like we do the genomics, like isolate the DNA or the chromatin, and then do specific sort of experiments depending upon the functional or structural genomics. Then we put them in the sequencing machine. Then once we get the sequence, you can see the magnifying lens. You get the sequence, and most of the time, even if doing the functional genomics, you end up getting sequences. Even for RNA or for epigenetic mechanisms, you end up getting sequences. And that's how the output of the almost all the genomics experiment, you get a sequence. Okay, then it's a computational analysis. We need to use the computer, otherwise it's not easy, because we are talking about billions of sequence, not like a few. Then we have dedicated people to work on, and then based on the result, we will ask specific questions on the epigenetic mechanisms that drive the tumor, or we have the drug experimentation, drug screening project as well. So this is what basically we do. And as I was mentioning that, just to give an example, we study sarcomas, like, which is mostly for happening in adults, sorry, not in children or young adults, like teenage age group, like Ewing sarcoma. Then for children, neuroblastoma, and there are different types of tumors that we study in our lab. Next slide, please. This is just an outline. You don't need to worry about the technical terms. But in 2003, we had the Human Genome Project finished, not completely, but still, we call that we have the complete genome information by 2003, just by sequencing the entire genome. Then there are other techniques that I have highlighted, that trying to understand the modifications of the histones. We use a technique called chip sequencing. Don't worry about that now. Or there are different types of techniques being developed over the period of the time that we have right now. We have highly advanced sequencing machines and techniques which can be utilized for the genomics to drive the structural or functional part of the genome. Okay, there are different based on the modifications, the methylation of the DNA, or the modifications on the histones, or the accessibility of the DNA. There are different sorts of techniques that we can utilize, that you guys will learn and study when you move forward with your masters and PhD and so on, if you're interested in the research field. So just to give a nutshell of this, these are some of the few techniques that we use in our lab to understand the genome biology. And next slide, I think I'm done almost. Yes. Just to put things together, this is our lab and collaborators, and then we have our bioinformatics team that work, and we have few colleagues. Not everybody is in the picture, but that's our institute, and that's the team. So, and thank you for your attention. I hope it was a little bit faster, I know, because of the initial delay. I'm extremely sorry for that. And if you have any questions, how are we going to communicate again, Nigel?
Thank you, sir. Thank you for giving your valuable practical insights on advanced genomic technologies in healthcare. It was actually a thought-provoking session, and we get to know more about the genomics, functional, structural genomics, and also other aspects related to genomics. Thank you so much, sir.
Sure, no problem.
Now,
Yeah, if you have any questions, yeah.
okay, I'll request the participants to ask questions regarding the topic. If you have any doubts, please ask. You can also comment in the chat box. Participants, if you have any questions, please ask. You can also comment in the chat box. Sir, I have a question.
Sure.
Uh, Actually, Shub Hospital is actually a leading hospital in Switzerland.
Yes.
So, sir, can you please tell any recent research work going on in the hospital related to genomics?
Sorry, in between, can you repeat it?
Uh, can you tell about the any recent research work going on in Shub Hospital related to cancer or genomics?
I mean, that that's our group actually. So we were other than our group, let me see. There are like clinical research groups in the hospital, inside the hospital, which means they are mostly working on patients, data collection, and mostly clinical research. The basic research, there are only very few labs. And one of the labs is ours, and the next door lab is working on neuroblastoma, another pediatric tumor, and yeah, and there are drug experiment, like the real clinical phase trials and other clinical experimentation are also happening. But I don't have the details of those things. But yeah, but we don't have a lot of labs who are working on the cancer genomics. It's like one or two here.
Thank,
But we have university, which is another place. But yeah.
thank you, sir.
Okay. So if you guys don't have any other question, how are we gonna conclude?
Uh, Hello.
Hello. Yes.
Yeah. Okay. Yeah. How do you study this genomic changes from the next-gen sequencing? Is it will be reflected in the next-gen sequencing?
Yes. So in the chips, that's mostly using chip sequencing actually.
Oh,
So what you do, I hope you know chipping. So we use a specific antibody for example, if you want to study the methylation or an active histone modification versus a repressive or inhibitory crystal modification, we have antibodies for each and every modification. Okay, histone modification. Then we isolate the chromatin of the cell. Then we use the pull down, just antibody pull down of the using the specific antibody. Then we have the ability to what do you call chop the DNA and then put it in the sequencing machine after library preparation. I don't know how well you're trained, so this is the reason I'm trying to use the technical terms. But the very simplest way you can say that, okay, self-example, if you can active antibody and repressive antibody, we use two tubes to pull down the chromatin associated with that mark. So we have only the specific chromatin which has that antibody bound. Okay, then we purify, we remove all the histones and all the proteins, and we end up with the sequence, just the DNA. Then we prepare the library and put the DNA in the sequencing machine. And when you sequence it, you get only DNA which had that modification. Okay, then we go back to the complete genome and align the sequence and say like, okay, these are the genes or these are the regions which had this modification compared to the other one. So this is how we study the epigenetics. Like otherwise, um, you know, uh, as I was mentioning, almost all the end reading is sequencing. But the before what you do is the critical thing, before the sequencing experiment, you do certain types of experiment technique to select the modifications
but
or the questions we're asking. So it's yeah, that's most of the time it's chip sequencing. Okay. I hope I was not super technical or
no, no, it was
specific.
very
I Okay. So yeah, I mean, you can anybody if interested in talking about this one or getting to know about more of our research or the research in Shu or Switzerland, you guys can contact me anytime, not a problem. And you have my
I'll be giving
contact information.
your email address. Is that fine?
Yeah, sure, no problem. Yeah. Anybody who have any specific questions and or the research options or things like that, please contact me. Not a problem. Okay. And I wish you
Yeah.
all the best and hope everybody is safe home with the situation, and hopefully things will get better, maybe in months or maybe in a year. So, okay. So thank you for all, and have a nice evening. Bye.
Yeah, we have a formal vote of thanks.
Oh, really? Okay. Thank you.
Respected Principal, Dr. Sister Asha, a most valued resource person of the day, Dr. Sanal Kumar Sir, Head of the Department of Biotechnology, Dr. Vici Mary Verghese, Director of Research Center, Dr. Sister Floret, faculty and students of St. Joseph's College, and participants from other institutions. It's my privilege to propose a vote of thanks on this occasion. On behalf of Research Center and Department of Biotechnology, St. Joseph's College, Autonomous Irinjalakuda, and my own behalf, extend a very hearty vote of thanks to Sanal Kumar sir for his effort towards this webinar. Thank you, sir, for sharing with us your works and findings and making this webinar fruitful. Further, we are very much grateful to our beloved principal, Dr. Sister Asha, for being the backbone of our every success. I must mention a deep sense of appreciation for Dr. Sister Floret, Director, Research Center, for her efforts to make this webinar a success. I would like to express my sincere thanks to our HOD, Dr. Vijay Mary ma'am, for providing encouragement. Thank you, ma'am. I also extend my special thanks to Dr. Nigel George for providing us such an eminent and vibrant speaker of today. Thank you, sir. We have been fortunate enough to be backed by a team of very motivated and dedicated colleagues. Thank you all for making this event a grand success. Lastly, I thank all the participants for being with us this evening and making the webinar fruitful. It's been a great pleasure. Thank you very much.
Sanal sir, thank you very much. I know you are very busy in research and everything. And because I think it will be your lunchtime now, right?
Yeah, I have some more experiments to do before.
Okay.
It's like 12. Yes, it's 12 o'clock. Sure, no problem. Okay, keep in touch.
Thank you so
and bye. Sure, no problem. Bye.
Once again, I am thanking everyone present and involvement in the webinar. It was a grand success. Thank you everyone.
നിങ്ങൾ കുറച്ചും കൂടിയും ക്വസ്റ്റ്യൻസ് ഒക്കെ ചോദിക്കണ്ടായിരുന്നട്ടാ. നിങ്ങൾ എന്തായാലും ചോദിക്കാൻ ക്വസ്റ്റ്യൻസ് ഒന്നും. എവിടെ ജെനറ്റിക്സ് പഠിച്ച കുട്ടികളൊക്കെ എവിടെ? എപ്പിജെനറ്റിക്സ് ഒക്കെ പറഞ്ഞിട്ട് ആരും ക്വസ്റ്റ്യൻ ചോദിക്കണല്ലോ.
അതെ, എല്ലാവരും മടുത്തു തുടങ്ങി, അതാണ്. എങ്ങനെയെങ്കിലും പോയാ മതി. ആ ചേട്ടനല്ല പോണത്, നമ്മുടെ ആളുകൾക്കേ തോന്നി.